Yeah, it's my great pleasure to introduce Giovanni today. Giovanni is a PhD student in the bioengineering program. He actually has defended his dissertation and submitted his dissertation already last month. His talk today is going to be about a project, one of his many projects that he worked on during his PhD. And it's about a single-cell RNA-Seq analysis and a new method that he developed to deal with single-cell RNA-Seq data. Okay. Thank you all for coming.
Okay. Thank you, Dr. Xiao, for your introduction. I'm going to go ahead and share my screen. I just want to make sure everybody can see the right screen.
Yep.
Yeah. Good afternoon, everyone. Today, I'll present a novel method that we've developed. The name of this method is scAllele, and it is a tool designed for the detection and analysis of variants in single-cell RNA sequencing. So I would like to start by introducing single-cell RNA sequencing. This type of data captures the transcriptome of individual cells, really giving us a complete glimpse of the RNAs present in every individual cell. And with this type of data, there is plenty of opportunities. The most clear one, of course, is that it allows for the characterization of unique cells or groups of similar cells. It offers a complete glimpse into the cell's transcriptome. And to this day, there is a really large body of data that is publicly available for analysis. At the same time, because it's a new technology, there is also certain challenges, such as probably the main one, the fact that this type of data is usually very shallowly sequenced. Although there's been a lot of studies done on this type of data, most of them have focused mostly on the study of gene expression. And to this day, there is no suitable method that is specific or tailored for variant calling in this specific type of data. With this in mind, we've developed a novel tool called scAllele. scAllele is both a variant caller and a variant analysis tool. It calls nucleotide variants in the single cell data via local reassembly. It's highly accurate and sensitive, especially in low coverage regions. And the method that scAllele uses in the detection of variant also allows for their functional analysis. The most important feature of this, of RNA sequencing in general, and that scAllele makes use of, is that the reads really capture a lot of information in the transcriptome. So inside a cell, we may have multiple transcripts, RNA transcripts. And these transcripts, of course, result from the transcription of RNA molecules, which contain the sequence information, but also it contains information about the processing and maturation of this RNA, such as the splicing isoform. And these different types of processing will lead to potentially different protein products that will also in turn result in different traits. RNA sequencing captures this diversity of transcripts. And what we can observe in the reads is that the sequence information and also the evidence of RNA processes such as splicing is also captured in the RNA sequencing reads or the single-cell RNA sequencing reads. And our method exploits this fact and performs these two tasks, which is the variant calling, which is identifying nucleotide differences in the reads, and also allele-specific alternative splicing, which is finding the association between alleles of a variant with different forms of RNA processing. In this case, different forms of splicing. I'm going to quickly outline the algorithm of scAllele. We start by parsing the sequencing reads from the single-cell RNA sequencing data. And we start by identifying these regions that we call read clusters. And read clusters really consist on regions of overlapping read segments. And these read clusters are the unit in which we focus in scAllele focus on one read clusters at a time. And within each read cluster, we search for the presence of variants. And we also look at the flanking introns in order to find the alternative splicing events. More specifically, we start with the sequencing reads, the sequencing data, and we also obtain, we create this sort of reference read, which is just the reference sequence of the interval where the reads are aligned to. And this reference sequence is used as an additional read into our reassembly. We go ahead and decompose these sequencing reads into k-mers. Here shown every k-mer as a rectangle of a different color. And we then reassemble these k-mers into a deBruin graph. This deBruin graph contains structures that tell us information about the sequence in the reads. For example, this bubble structure shown over here represents sequence differences between the reads, and these sequence differences are in fact variants. For this, the identification of the nodes that are present in this deBruin graph also give us information about what nodes, what specific sequences mark the beginning and the end of the variant position in the graph. And in this presentation, we can further simplify this bubble structure into this symbol over here. A typical read cluster, of course, includes multiple variants. And depending on the complexity or the type of sequence, the overall deBruin graph that is assembled can result in multiple or even nested bubble structures and cyclic structures. The overall goal is to find a path in this graph that best represents the original sequence of the read. For every read that overlap the read cluster. The problem being, of course, is that depending on the complexity of the graph, depending on the cycles present, there could potentially be multiple walks on this graph that represent the original read sequence. And so what scAllele does is to obtain the walk with the smallest edit distance in this graph, the edit distance being pretty much the nucleotide differences between the read sequence and the reference sequence. And we assume that the combination of these differences with the smallest edit distance is the most likely original sequence of every read. And we do this assisted by a Dijkstra-like algorithm that will help us find this shortest distance in the graph. And what we end up with is a read walk, which is a walk in the graph corresponding to every read. And this read walk contains information about the variants present in that read and the alleles for that variant for every specific read. So what we end up with is this data that for every read, we have the allele information at every variant position. And this format allows us for the dual analysis of scAllele. The first one being identifying the variants, the variant calling part, which we already know the variant and the allele from the previous steps. So we go ahead and score them in order to find how confident we feel about every variant that was found. In order to score the variants, we use a generalized linear model that makes use of multiple features that are present in the data. Now, going a little bit into detail of these features, some of them are commonly used by other variant callers as well, and some others are some that we did our own implementation. Some of these features are the base quality of the read, the base quality of the bases at the variant position, the presence and the count of tandem repeats neighboring the variant position, the position of the variant on the read, and another feature is the overall sequencing error rate. These features, of course, tell us information about how likely the variant is to be a sequencing error, given that variants that have low base quality are surrounded by multiple tandem or homopolymer repeats or are at the edge of the reads would tend to more likely be a sequencing error. And this constant gives us the overall expectation of finding an error in the reads. We use another feature that was of our own implementation here, which is given the individual reads and the alleles that contain individual variants, for a diploid organism, we should be able to cluster this combination, these haplotypes, into two haplotypes. And this filter is called haplotype fitting. So failure to cluster the haplotypes will likely indicate that there is one or more variant that is the result of an error, false positive variant. And also another feature that we look at is the allelic balance or allelic ratio, which simply refers to the ratio of reads that contain the alternative allele of the variant from the total number of reads that overlap the variant position. And this feature also tells us information about the variant, because a variant that is present in a very small fraction of the reads would potentially also be a sequencing error, or practically in this hard to distinguish from a sequencing error. We went ahead and evaluated the performance of scAllele, for which we obtained single-cell RNA sequencing data from the GM1287A cell line. And the way the metric we used for our evaluation was the count of true positive variants detected, and which would be the sensitivity of the variant caller at given false positive cutoffs, which is we only allow this many false positives. And as you can see, we compare our method with three other methods that were previously used in single-cell RNA sequencing data and are commonly used also for other types of data sets. And we show that the panels, the top panel here indicates performance on microindels, and the lower panel indicates performance on SNPs. And we show that the performance of our method for SNPs first is similar compared to the other commonly used methods, but the performance on microindel is clearly superior compared to the other three methods. And this result is consistent if we consider different benchmarks. For this set of results, we use benchmark from the Genome InnoBattle that sequenced the genotype of this cell line. But if we use other two sets of benchmarks, this is the set of variants for this cell line that are considered high confidence. And this is the set of variants that were obtained using third-generation sequencing. We see that our method consistently performs similarly for SNPs, but consistently outperforms all the other methods in microindels. And this is because during the development of this method, we had the priority to identify microindels with high accuracy because microindels are more notorious for their difficult identification. We perform other types of evaluations as well. Here we have the similar type of analysis, except that we focus on difficult regions, variants located on difficult regions. And difficult regions refers to regions that are hard to map, that contain repeat sequences or segment duplication, high GC content, etc. Overall, regions where variant calling is hard to perform. And we also show a similar trend in the results where our performance on the detection of microindels is still superior compared to the other three methods. And going back to one of the points mentioned in the motivation for this project and this type of data set, which is that in single-cell RNA sequencing, most genes are shallowly covered. In fact, we find that the majority of variants, up to 95%, are very lowly covered, which means they contain 5 or less reads. So it's very important to have a method that can call variants without needing a whole bunch of reads. And we show that for the variants that are very lowly covered, which would be the left hand side of this graph, our method, scAllele, has the highest performance on this range. And we can see the differences also, again, particularly noticeable for the detection of microindels, where microindels with this many reads, where scAllele would detect more microindels that are covered by this many reads. So that covered the first part of the method, which is the variant calling. The second part, which deals with the functional analysis of the variants identified, also makes use of this data format. But in this case, we aim to find linkages between pairs of variants. And the linkage refers to the co-occurrence of alleles, of the alleles of two variants. For example, for variant 1 over here, whenever we observe an alternative allele, we may, for example, consistently observe the reference allele for variant 2. And the way we evaluate this and quantify this is by calculating the mutual information between variants. And for the mutual information, we consider the two random variables that we're testing as the alleles of the two variants. And this way we capture the information entropy that we can gain from one variant on the other one. We can furthermore expand on this idea because we're calculating the linkage, the co-occurrence of alleles, but we can also expand this idea to splicing. And the way we do this would be to consider different splicing isoforms as alleles of the same variant. So what that looks like is, for example, if we have reads that contain introns, so two different introns that represent two types of splicing, as long as they overlap, we can consider this common region as the variant and the two types of introns as the two alleles of this variant. So doing this conversion, we can also find linkage between a genetic variant, which may be located somewhere upstream or downstream. We can also find the linkage between the alleles of the genetic variant and the splicing variant, if you will, in order to find association between variants and splicing. We apply our method scAllele to detect these allele-specific alternative splicing events in lung cancer. The lung cancer dataset that we used was sequenced using SMART-seq2 dataset, and we had both cancer and normal cells. Here we show the number of events, the number of ASAS events that we found in the normal and cancer cells and in both of them. And we consistently observed that there were more events present in the cancer cells, more allele-specific alternative splicing events in cancer cells. And we also observed that many of these ASAS events are also very specific to individual cells. If we show, here I'm showing the prevalence of events and the number of cells that they show up in. And we see that the vast majority of events are present in a few number of cells, showing this high cellular specificity. What these events look like, in order to illustrate, here is one example of an ASAS event located in the BCL6 gene. And here we have split the sequencing reads by the allele they contain on the variant position. So we have a SNP located over here, and we split the reads based on the allele they contain on the SNP position. And we can see that the reads that contain the C allele would tend to map to this middle exon, and the reads that contain the G allele would skip this middle exon. So this is a very clean example of a allele-specific alternative splicing event. And we can furthermore try to find differential ASAS events, given that we have two conditions, cancer and normal. So here we report the number of events and the category of differential that we define. We can consider differential events if they're only present in cancer or only present in the normal sample, or if they are present in both, but they are more prevalent in the normal sample. So we'll name that one normal differential. We didn't find the missing category, which would be cancer differential. Those be the ASAS events present in both, but mostly enriched in cancer. And we also show the number of cells that contain these events. And just to illustrate this idea, here we also have another selected example of a differential event, in this case in the CTSE gene. And here we also show the sashimi, the allele-specific sashimi plots for this region in a normal and a cancer cell. And what it shows here is that for the normal cell, the splicing of the reads containing one allele, in this case the C allele, is different from the splicing of the reads containing the T allele. But this difference is only present in the normal cell. In the cancer cell, the same gene, we observe that both alleles splice similarly on the same exon. So this event is only ASAS in normal, but it's not ASAS in cancer. So this would be considered a differential ASAS event. And in summary, our method, scAllele, calls single nucleotide variants and microindels with higher precision than other commonly used tools. The variant calls made by scAllele are sensitive to log coverage and also difficult regions. This tool also allows for the discovery of allele-specific alternative splicing event. And when applied on this lung cancer data set, we identify over 150 ASAS events and several differential ones across the two conditions. And with that, I would just like to thank my advisor, Dr. Grace Xiao, and also all my lab mates in the Xiao lab, and all of you for listening.